Hello and welcome to the second episode of Fungi Files. Tonight we're going to be discussing the ochraceocentrata paper. This is a preprint currently titled Discovery of the Closest Free Living Relative of the Domesticated Magic Mushroom Psilocybe cubensis in Africa. And tonight, discussion panel we're going to be doing this with is Dr. Jason Slott, as well as Julian Mattucci and Scott Otsuni. So I want to give each of our guests here a chance just to introduce themselves. So thank you for joining us tonight, and I'll open the floor up for you guys.
I'm Jason Slott. I'm a fungal evolutionary ecologist at Ohio State, and I am not in any way representing my employer, especially when I entertain outlandish ideas, which sometimes happens.
I'm Scott Otsuni. I'm a sequence validator for the Fungal Diversity Survey, the Ohio Mushroom DNA Lab, and Mycota Labs. Um, yeah.
Thanks, Scott.
I'm Julian Mattucci. I'm the owner of Imperial Labs and Imperial Scientific, and I run the Imperial Labs Culture Bank.
Thanks for joining us, Julian. I've had plenty of conversations. I know my audience knows you quite well.
Yeah.
Well, this paper was, it's been on everybody's lips, I'd say, in the community for the most part. There's been a lot of talk about it, and some agreement, some disagreement, as is expected, I suppose. But tonight, we just wanted to get together and go through the paper and have a discussion, kind of a little friendly peer review, and see what our friends here have to say about it. I don't know if you want to read any of this, Dr. Slott, or if you have any points you'd like to bring up here at the beginning before we kind of dive into it.
Uh, I'm thinking kind of free form, but I kind of just wanted to give a quick overview of the main methods here. So this is a combination of a barcoding and genome sequencing study. They sampled mushrooms from Africa that had been named cubensis, Psilocybe cubensis, and they've done some phylogenetics to kind of tease apart the relationships amongst those mushrooms, and they've done some molecular dating to put it in an evolutionary context, trying to figure out where Psilocybe cubensis and this new species, which isn't necessarily all that new to the Psilocybe community. It's seen it going around for a while under different names. And then they did some climate modeling for species distribution as part of their evolutionary analysis. And this is also a taxonomy paper, and they've done some nice microscopy to define this provisionally named species, Psilocybe ochraceocentrata. Did I get all that right?
Sounds right to me. Thank you. There's a lot of interesting points in the paper to go over, but yeah, it's definitely a nice overview of kind of the protocols and stuff that they used here. I think that's probably one place to begin. Actually, is some of these methods used, like the ecological niche modeling. Do you guys have any opinions regarding those sorts of methods?
I mean, to be honest, that's above my head personally. So I don't have any opinion on that.
Sure.
Yeah, I can't really comment on it, I would say. I understand what they're saying, but I can't review it and say, oh, yeah, they did a good job or a bad job with this. I don't understand really how they're doing it, but I understand what they're saying.
Yeah, the reason I ask is because I know there's like, when you talk about things with like molecular clock and things like that, there is some, um, it's not always agreed upon everybody that's like 100% accurate. So it's kind of why I bring it up. Um, But as you say, I'm also not not versed well enough to really speak on it. It's just something I'm curious about.
Yeah. The thing about molecular clock, I mean, is it can be inaccurate based off the phylogeny, but the one thing that I've noticed is, if you have pretty good consensus on those numbers, that's still most likely what happened and roughly maybe when it did happen. There could be, some discrepancy there still, and we have incomplete phylogeny. So there'll
Hmm.
always be a little bit of guessing. Um, but yeah, I don't think I don't think they would be too far off with their 1.5 million year divergence with the molecular clock analysis. I mean, maybe it's a million, maybe it's 2 million, but there
Hmm.
is a major divergence a long time ago, and I think that's accurate enough.
Well, maybe you could bring up figure 1, which kind of puts together the sampling and the phylogenetic tree, and the molecular dating analysis. You have that one handy?
Yeah, sure. Give me just a second here to pull that up.
So most of the results that speak to me are in that figure. And I'm not necessarily saying that I agree with it all, but they're speaking my language.
So yeah, here's figure one. Yeah. So what on here did you specifically want to speak to, Dr. Slot?
Yeah, I'm gonna take a little bit bigger picture of this. Just to arrange my thingy here. So Ochraceocentrata is right here, and they're showing several specimens. I believe they found 6 across Africa. Well, actually, mainly in southern Africa. And then there's a sister relationship that they're showing to Psilocybe cubensis. And one important feature of this story is that everything that was named cubensis previously, or some of the older alternative names for cubensis in Africa, are falling into Ochraceocentrata. So those are colored differently here. These South Africa and Zimbabwe accessions than the cubensis, which is the uh, reports are in this, I don't know what that color is, gold
Gold.
across North and South America and Australia. And so here's the first dating result that they're reporting is this 1.56 million years ago. And it's kind of cramped in there. You can't see that error bar in there, but those error bars are all pretty big. And this one is supposed to be what they're calling the highest posterior distribution. Well, the Bayesian analysis is at 1.56 million years, but they're really covering an estimate of 0.71 to 2.5. From the perspective of this entire tree, that's actually not a big difference. It's pretty much yesterday. But I think kind of one of the important questions that we have is uh, things that might have happened in the last 500 years. So I don't know that this tree is really going to be able to address anything in human history, well, in modern human history, and definitely not in recent human history. Uh, and then they've also put some other taxa that were formerly labeled as cubensis, like the Psilocybe aquamarina was thought to be a subspecies of cubensis, I believe. Does anybody remember that from the text?
Believe
And
it
I'm not
was
sure where that's
potentially
from.
synonymous, but yeah, it's like they turns out that it was an
Closer to
independent
Nivea and
species.
stuff. I remember
Yes.
someone was saying it wasn't even a psilocybe, that it was like a Deconica that was like misidentified or something along those lines, or maybe a Stropharia that was misidentified. But yeah, it looks like, because I talked to them about that, and they said they used the holotype for Aquamarina, so it's obviously some sort of Psilocybe closely related to Nivea tropicalis.
Yeah. One other little tidbit I noticed as I was looking at this this tree was that they used sempervive instead of subtropicalis. So I'm wondering if that's going to if subtropicalis is going to go away and upset some people.
It depends what sequence they use. Is it in Mexicana or Zap?
Here they have it.
Zapotec.
Yeah.
Okay. Yeah, because there's this sempervive sequence on GenBank that's a Hochscheggenii. So it'll go in Mexicana if you like make a tree out of it.
Mm.
And it's confused a few people, I know that.
Yeah, this one has Hochscheggenii down next to Mexicana.
Oh yeah, nice.
You can also look at the big picture of the dating analysis that they've done here. First of all, they based their entire analysis on one calibration point. So they took a date of 67.6 plus or minus 6 million years, or I believe it was this node right here, the origin of Psilocybe. Actually, no, according to this, it would be the divergence of Agrocybe and Psilocybe. And so here's the 67 million years, and then this bar is the error bar even around that. So you can see that these error bars are covering quite a bit of territory. So some of the things that they're showing are this, I think this is the Eocene climate optimum and the Middle Miocene climate optimum, and they're corresponding to maybe some major events like the divergence of clade 1 and clade 2 Psilocybe.
Mm-hmm.
I was curious with this one why they have clade 1 and clade 2 split the way they do, because like in the Bradshaw paper, clade the clades are flip-flopped.
Yeah, that's an error. So
Yeah.
oh, wait a minute. No, hold on. Am I getting confused? Yeah, clade 1 will have the type, which is
Semilanceata.
semilanceata.
Yeah, which is in their clade too.
Right. It's actually not lined up right here either. It's
Yeah.
sort of roping in Carolensis and Subcerulipes. Yeah, preprint.
Because my understanding, the Mexicanae and Zapotecorum are clade 1 species. And then cubensis and Lanodensis niviotropicalis, Psilocybe cyanescens, Aztecorum, all those are clade 2. So yeah, it's like that's backwards on that.
Yeah, that's an oversight. I'm sure that will be picked up in review. Anybody remember the deal with Congolensis? Clearly it's from Africa, but I'm not sure it was ever called a cubensis. Or they just...
No, Congolensis I thought was pretty close to Mexicanae. Oh yeah, there it is.
Yeah, I'm just wondering why they've highlighted
Why
it.
they have it bold.
Yeah.
Yeah.
Was Carolensis also Africa?
Carolensis was supposed to be from Kerala, India. But
Yeah.
that one's really weird. Because the spores that I've seen for what's supposed to be the type look like Cordyspore spores. And then there's multiple sequences on GenBank, and I don't know which one's right, but one of them matches a specimen that people are calling Thai Zapoteca. But I thought Thai Zapoteca was synonymized with Argentipes. So yeah, the carolensis is a weird one for me.
Seems to be widely distributed as well, like all over Asia, which is
Oh, you know
probably.
what? Maybe if it's in, or no, that wouldn't be right.
I'm hoping to get a little more clarity on this Ties thing. I have some genetics from India that are producing pins, so hopefully we'll have fruits to look
Yeah,
at.
that's the thing that people are calling Ties apotheca.
Right.
That
That's
at
what I
first
mean.
people were calling carolensis, but I don't think it's either of those.
Mm-hmm.
Or well, I think it's maybe Ties apotheca, but like I said, I think that might be synonymized, but I don't know if that was correctly synonymized either. So,
Yeah. Well, it has features that look like Zapticorum from the pictures I've seen of the fruits, so not sure what to think.
well, there's pictures of fruits that look completely like a cordyspore, and there's pictures of other fruits that look like sections apicorum. And I
Hmm.
don't know if it's just, morphological variation there, or if people are finding some sort of sections apicorum species and calling it carolensis because it is in Kerala, India.
Yeah.
Because that's why we were calling the ochraceocentrata natalensis, because it was found in Natal, South Africa. It
Sure.
looked pretty much similar to the description of natalensis, and no one was guessing that it was just another species on another cubensis-like species in South Africa in Natal.
Right.
So that seemed the most likely thing.
Yeah.
Yeah. It also highlights the issue of like naming a species after the type locality. I mean, what if we end up finding Mexicana in Tampa, Florida, where that'll be a shit show.
Do you guys know how a type, is chosen?
It's up to the author. Ideally, you'd want a good collection of it with as many fruit bodies as possible. Historically, it probably would have looked a little different. Because historically, a lot of people named a species after a single collection, and that might have just been like a single fruit body, um, which is unfortunate because then it limits your ability to study it. But I mean, like for Nivea tropicalis, I decided to use the first collection I made, which it definitely doesn't have to be that. But it was a big collection. I didn't have the greatest photos, but yeah, it's kind of up to the author. But ideally, you want a large collection so it's easier to study and you can, describe it based on like multiple specimens within the collection as well as other collections, which and turn into pair type collections. Does that answer the question?
Yeah, it does. Thank you. Yeah,
Okay.
as I was
Yeah.
reading this, I was just thinking like, who chooses the type specimen? You know, like,
Yeah, it's whoever the author is or the authors on the paper that describes it. But it has to be accessioned into an herbarium and annotated as a type specimen.
hmm,
Yeah.
cool. Thank you.
Hmm.
I think it would have been nice if they had put the geography on here, kind of just to be able to think about the biogeography of its distribution. Because the main point of the paper is that you've got kind of the center of diversity for part of the cubensis clade in Africa. And it's kind of the majority of the paper is based around that whole story.
Yeah.
Right.
But cubensis has actually made it out.
Yeah, the one thing I thought was interesting is they said after like 2 years of surveys throughout South Africa, in Zimbabwe, in Zambia. They didn't find anything that sequenced as cubensis. So it's like they haven't found a cubensis collection in South Africa. The researchers, at least. I'm not saying it's not possibly there. But...
Mm-hmm.
Well, another thing, I don't know how prevalent the cultivation of cubensis is in South
Mm-hmm.
Africa, but if they do find it, who's to say it wasn't introduced through cultivation,
Exactly.
kind of like the
Yeah.
gold here. Um, I think about that often with a lot of these landscaping philosophies. Let's say tomorrow someone finds a lenni. I don't know, has a lenni been found like in a natural habitat? I know cyanescens has. I'll use that but it's like, was it already in the forest and then did it hop onto the mulch, or did it go from the mulch to the forest? And, you know,
Well,
I don't know,
population genetics actually
addresses
tells
that.
us this.
Right. Do we have that kind of data on some of these wood lovers?
So Alistair McTaggart did a lot of that work for Subaruginosa, and he placed Alanii and cyanescens and azurescens into a more complicated Subaruginosa, whose center of origin is in Australia,
Okay, that's right.
and the center of diversity is there. It's kind of how you can infer where it came from and that it dispersed.
That's interesting though, because in New Zealand, aren't all those species present there, as well as Macaroa and Wararoa, which would mean New Zealand has like higher diversity?
Good question. I don't know where Wararoa falls in the phylogeny. You know of a paper that
Its
includes it?
ITS sequence is similar to cyanescens, or the same as cyanescens, but they said they found none in Australia. When I talked to Kane about it, he said they didn't find any Wararoa there.
Yeah, so
But yeah, I thought that was interesting.
if it's actually in
That like group of
Subaruginosa,
species. Hmm.
then I don't know if you can say that that means that it's more diverse in New Zealand, because if Wararoa is just a population of Subaruginosa, is it really more diverse because we gave it a different name and it's got a shape that's different? I don't know.
Yeah, I just mean like related species. Subaruginosa, if all those things fall under Subaruginosa and their Subaruginosa is found in New Zealand, but Macaroa and Wararoa are there, then there would be like more diversity of that group of species. Especially because I'm potentially, with the logic used in the Australian paper, if Wararoa has the same ITS sequence as cyanescens and mates with all of those species, then it would also fall under Subaruginosa, and it's not in Australia. So
Yeah, but how do you call diversity, right? Is diversity at the genomic level, or is diversity at the morphological level? As if,
well, I would think just species wise, because like Macaroa is obviously not Subaruginosa, but it's a very closely related species to Subaruginosa, and you don't find that in Australia either. So it's like if you find all these similar species in New Zealand as well as Subaruginosa, I would think that that's where the more of the diversity is. But I
well,
think
definitely,
that
yeah, if you have
maybe
a sister
maybe that's why
species.
they say Oceania
Hmm.
instead of Australia, because I guess New Zealand's part of Oceania.
What did you all think of the niche modeling? Let me pull up the figure 2 niche modeling. So they picked three date ranges, I guess, to model with two different types of modeling: ecological niche modeling and species distribution modeling. I really don't know what's under the hood and how those two things differ.
Hmm.
Yeah, I couldn't tell you exactly. I mean, I know they were looking at like climate patterns and stuff like that and trying to model out where these species would occur throughout time based on climatic modeling, basically. I was curious if this is like taking into account like continental drift and whatnot, because the amount of time we're talking about is massive, and obviously the maps we're looking at are like the same. So I was kind of curious about if their modeling takes any account to the
Well,
fact
I
that
thought
these
for
things would have been
continental
all over,
drift,
right?
it happened so long ago that because if Psilocybe started 65 million years ago, then that would have been well after the continents split up.
Okay. Yeah, I was just thinking back like 70 million years. I guess I was thinking on the wrong time scale.
So the Isthmus of Panama formed between 3 and 23 million years ago. So it's just really fine tuning on these continent movements,
Mm.
although that's a pretty important one,
Yeah.
the formation of the isthmus. But these are, yeah, 3 million years is really not that much. A lot happened from our perspective, and we went through a recent major ice age and moved from the Pleistocene to modern. But I guess that they're saying that back here, or maybe more in the Pleistocene, Africa was more hospitable. This is what I'm getting from the text. Africa was more hospitable when the common ancestor of ochraceocentrata and cubensis was in Africa,
Yeah.
and then it moved out, and afterwards it became Africa kind of became inhospitable to cubensis, whereas the Americas became more hospitable and
Hmm.
Oceania became more hospitable, Southeast Asia. But I have a hard time really seeing that in this figure.
Hmm.
I think maybe there's some numbers that underlie this that you can go by. But
Yeah.
yeah, visually it's kind of hard. You could see some
Yeah.
changes in Brazil.
One thing Kyler brought up that was interesting with the Natalensis and the Ocraceocentrata is their occurrence at like that southern tip of Africa where the climate's kind of still stayed hospitable after Africa went through like it was all rainforest and then a large part of northern Africa became desert. I'm not sure when exactly that happened though either, but it seems like that's a possibility that Africa was more hospitable and then everything was kind of driven south as rainforest turned into desert in the northern part. But yeah, I don't really see that in this climate model.
Yeah, I guess down here in the Pliocene, there's a range of hospitability, and then it's really by the Pleistocene, there's a band across the center, but I guess it's still there in the contemporary.
Hmm.
Yeah.
Yeah, from the text, like I got more from the text than this figure.
Yeah.
It says like these major migration events present the possible avenue for dispersal of cubensis and ocratiocentrata from Africa, and subsequent divergence into Asia and Africa after aridification led to loss of habitat in the intervening region. So that makes sense to me. I don't quite see that here
Well, yeah,
necessarily either,
that's through
but
here, but
yeah.
I don't see that as really being represented by, I don't see any indication that it was going through the Arabia or
One thing that I thought was kind of interesting is they're talking a lot about like herbivore, like ecology and how that is like linked to these dung-loving mushrooms, and how like bison, like ecological engineers spreading grassland ecosystems where they roam because they knock down trees. And um, one of the other things that mentioned though also is that it's like there was the whole idea that cubensis was introduced to the Americas via cattle from the Spanish in like the 1500s or whatever year that is. Um, and this paper kind of seems to challenge that based on this the dating basically. Do you guys like? Does that make sense to you? Or
Yeah, they're suggesting that it's plausible. I don't think you can say anything about where Psilocybe cubensis originated based on the 1.5 million year date,
mm-hmm,
right? It clearly has spread throughout South Asia, and it is now throughout a lot of North and South America and the Antilles archipelago. But it it just puts an outer bound for when it would have emerged. But it really can't say anything because there's no historical specimens from the Americas. There's no sister species or closely related species in the Americas that would suggest that it did originate there.
mm-hmm,
But they have made this sort of speculative argument that once it got into Asia, once it left Africa and got into Asia, that it followed the migrating herbivores, well,
mm-hmm.
the ungulates basically, as they're talking about bovines, bison,
Yeah, that they may
across
have
the
transported them from Asia to the Americas.
yeah,
Yeah.
it traveled with them over the land bridge and into the Americas, and then they made a big argument about the the spread of bison and how Psilocybe cubensis is really sort of specialized on bovines, which maybe, and I sort of looked up like if that is really supported because I know that it has been isolated from hippopotamus, well, not hippopotamus, but elephant
Elephant dung.
and
Yeah, it's a,
horses and like the wild horses and proboscids and all that. So it is capable of using other dung. It doesn't necessarily have to be spreading with
yeah. Seems
bison
like
or domestic
any
cattle.
any grazing, like grass grazing dung producer, I think generally cubensis would probably eat that.
Yeah, as long as it's big enough, I guess, because they're a bigger mushroom. I don't think
Mm-hmm.
it would be. I don't think rabbit would be
Yeah.
a preferred.
Yeah, in the paper they say that they cannot rule out that America was an origin with the present data, but it's like the inferred divergence makes more sense from Africa. And I think the way
Yeah,
they said it was like it's the thing about it being brought from its ancestral home with the Spanish makes it like less parsimonious was the wording they used.
Yeah.
yeah, but I don't know that that's the necessarily the the inferred point of origin for if it did come from human migration. Like, why couldn't it have come in some direct or circuitous path from Asia, Southeast Asia? I mean, there was trade between the East Indies and probably not directly between East Indies and West Indies, but there were certainly cattle and horses on those boats that were going to Europe and then to India and then to the Caribbean islands. And
Hmm.
so I think 500 years ago is still on the table, especially when you compare it to Alistair's work
Hmm.
with the number of generations that he calculated for how long Psilocybe cubensis has been in Australia. Right? He calculated 75 generations, and it was based on an analysis of single nucleotide polymorphisms. But if you look at the single nucleotide polymorphisms in all of the strains that he sequenced, some of which came from Asia, I believe, and some of which came from the Americas, I believe, it's really just back of the envelope. It's really only about 10 times that. So if all of that diversity of cubensis that's in the combination of Australia and the commercial strains covers somewhere around 750 generations, that could line up with a dispersal event 500 years ago.
Hmm.
But I think that we don't have the data here. This is not the data set to test that question. Like you really need to sample genomes throughout the Antilles and South America and southern United States and Asia and just really see how much diversity there is so that you can get a handle on how long North America isolates have been separated from Asia isolates.
Yeah, that sounds like a big job, but one that would be worth doing.
Yeah. Hmm.
One thing the paper mentions a few times is the undersampling of Africa and the species that probably occur there that we don't know about. Um, when we talked to Alan, we asked him to just like guess at a number, and he said like maybe at least a dozen or more undescribed psilocybe there alone in the African continent. And I was thinking about it. It's this gigantic continent, and there's, well, we just had a couple new ones described, but prior to this was like what, four I think that we had. Very few for a continent that size. If you compare that to how many species we look at in the Americas, it's like
That's Florida.
yeah,
Florida has
exactly.
more species.
Exactly, so it's like, and Africa is enormous. So there's got to be just at least it seems like there should be a huge amount of diversity there that waiting to be discovered.
Mhm.
Um, what do you guys think like contributes to it being so heavily under studied?
I mean, legality wise too, there are a lot more strict. South Africa's extremely strict with like removing biological specimens and stuff like that.
Mm.
Another thing is I was talking to people in South Africa. They they're saying they don't really have much sequencing there either. Like they'll have to send it to a university sometimes to get it sequenced if that university is even interested, you
Mm-hmm.
know.
Yeah.
So that was one thing I'm talking about because I have extra supplies to run PCR. So maybe just getting some of those supplies to somebody who can run PCR in South Africa because these guys are showing me a lot of crazy pictures of a lot of crazy things and some stuff it's like cubensis and ochraceocentrata and analensis do look a lot alike. It could just be some other thing that's cubensis. Like even that aquamarina, it's described as very cubensis-like. Now the phylogeny puts it closer to Nivea tropicalis, but that's what I mean. There could be many, many things there that we're just collecting under other collections already, or things that I think Ingali was only found twice. And when it was published, it had only
Mm.
been found once,
Once.
you
Yeah.
know?
Yeah.
So, yeah, I
Yeah, and
think,
there's Maludi too.
yeah, just the lack of coverage of mycologists per square mile, I guess you could call it.
Mm.
There's not,
Mm-hmm.
I think the population in South Africa isn't crazy per square mile. It's probably pretty low. So you just have less people and I think somewhat lower access to technology too. So
Mm-hmm.
I think
Yeah.
that's part of it, but obviously not the whole picture.
Sure.
Yeah. I mean, I was gonna like I have a friend who runs and it's not South Africa. I forget where, but I have a friend who runs a spirulina farm, and he's like donated and established like a spirulina farm in one of these countries in Africa, and It's like people don't even have the luxury or liberty to like spend time studying these things if they're focused on just feeding themselves, you know?
Right. Yeah.
Yeah, but I would just go back to that whole band across the middle of Africa, which is a huge area, is just so deadly from giant chimpanzees to like
Hippopotamus.
Malaria.
hippopotamus.
That actually shocked me because Penniless tropicalis that was collected from hippopotamus dung in Tanzania, Africa, and I'm just thinking, you've got to be a lunatic.
Yeah, you're risking your life to find some mushrooms.
Yeah, taking your life in your hands for some actives.
Yeah,
Sounds like fun.
Alan would do it.
Macromorphology here. I don't know a lot about this, but I imagine you guys do.
So A is the holotype specimen, I believe. Is that right?
Yeah.
Yeah. When I was looking this earlier, the mushroom on the left definitely has like a lighter margin, but that one on the right, it's kind of lacking that some of the features that ochraceocentrata is named for.
This one here?
Yeah, I mean it's got the center, but the margin is
It just
it
seems like
doesn't
it's like
fade out
kind
to
of
white.
wet.
Yeah, maybe wet or
Hmm.
yeah, I suppose that could be. There was some interesting things. I know we're talking about macro stuff, but there's a few interesting microscopic things that I wanted to mention as well. Between that one and a few of the others, that there was no Pleurocystidia observed, um, and no Chylocystia observed in type collection, but there
Hmm.
was clear Chylocystia in two of the other specimen, and also the spore size seemed to be quite a bit smaller from the type specimen compared to. Let's see what it say. It's just that they're variable in size. So, like one of the specimens has much smaller spores, with few in extreme ranges. So, uh, it's just one thing to talk about, I guess. If you're looking, like in Gelly, for instance, and looking at one tiny collection, there could be a wide range of spore size potentially in a species.
Right.
Go ahead.
Yeah, I was gonna say this kind of goes back to what I said earlier about, and not that they only had one collection. They had what, three collections that they used to describe this. Um, historically, mycologists basing their description off a single collection and then, describing, any of these micro or even macro features, especially with the size. Um, a lot of species descriptions could probably be rewritten, and the ranges for size for almost any feature could probably be expanded pretty drastically.
Hmm.
Uh, if you were to use, let's say, like 20 or 30 or 100 collections to base it on versus like one. Um, yeah, that's all I had to say.
Yeah. So then I suppose the question should be posed like, is spore size, or, I mean, is that do you think a good identifying feature of species? If it
It
could be
can
so
be,
variable?
What are the rules
but
for how many
you're
fruit
taking.
bodies you have to count? I mean, how many fruit bodies you have to measure spores from, or how many spores you have to measure?
Yeah, so
30 is usually, right? 30 spores.
yeah, but I don't think there's a rule about how many different fruit bodies you need.
Yeah.
Um, you need
You
to
like
just
sample?
Hmm.
right.
Yeah.
Obviously, if you only have one, but is it
Right.
Hmm.
is it strongly recommended that you look at more than one of the collections?
Yeah.
Well, the reason
I
I'm
mean,
asking that too, Scott, is because we just got out of a different conversation where somebody said that spore size wasn't a valid identifying feature, and I was like, my reaction in my head was like, what are you talking about? But I guess as this conversation develops, I'm like, well, maybe there is something to that. And when he said that, I did think about what I had read in this paper because I just refreshed it, read it this morning, where it's talking about this variability in spore size. So I'm like, well, I don't know. I mean, historically, it's been very, I feel like, pretty important feature for identification. But now I'm like questioning everything.
Yeah, I think it depends what it is and how much range you have. Like there have been a couple times people sent me spores that they said were subtropicalis spores, and I looked at them under the microscope, and they were Zapotecorum size, and the monokaryons from them would not breed with subtropicalis monokaryons, but they would breed with Zapotecorum monokaryons. And it's like I've seen a lot of consistency there, but I don't think you can reliably use any one single feature by itself for identification. It's really when you're using multiple features to kind of suss these things out. It's like, can you identify a car by calling it a white car? That kind of doesn't describe the car enough. But if you know the make and model and you say it's white, now we're a little closer. Now you know the license plate. So there's different features that once they line up, that's how you get the identification. And one feature on its own, same thing with ITS. Some things have the same ITS. You use ITS alone, you might get some confusion. You use spore size alone, you might get some confusion. But I think, like Scott said, we could have more robust collections and more robust data for the overall ranges and what those outliers can look like. But I think when we take into account the different things that we use for identification, then spore size matters within that context, if that makes sense.
No, yeah, that
Yeah.
makes perfect sense.
That, and I'll add one more thing. So I feel like it is informative in the context of breadth of the spore size. So like, all right, what do we got for spore size? Well, all right, right now I'm looking at basidia size. No, here, spores. Okay, so at one extreme for length, they've got 12 and a half, and then all right, so they've got 11 and 12 and a half for the lower and upper extremes, and then for the length, and then 6.5 by 8 for the width. So there's a difference between a spore that's 12 and a half in length by 6.5 in width versus 12 and a half in length and 8 in width. That's going to drastically change the shape of the spore. Um, and usually, the bigger the length, the bigger the width. So I feel like the breadth of it is more informative, or the Q value.
Mm.
Um,
Mm-hmm.
because like, you could imagine 12 and a half in length by 6.5 in width. That's almost twice the length as width, and that's going to produce a significantly different shaped spore, if that makes sense.
It
Like
does.
it's
Yeah,
going to be
that's a
more
good point.
elongated,
Mm-hmm.
right? Because like with like Niveo, I noticed this with Niveo tropicalis and Wayanadensis. There's a lot of overlap in the range of length and width, but when you correspond the length with the width, it's directly proportional, and produces a different size in the breadth of the spore. And, when you look at the data as a whole and the spores, Niveo tropicalis has wider on average spores than Wayanadensis, making it more like not quite globose, but closer to that end of the extreme.
Mm-hmm.
So I feel like that's where the most informative part of the measurements is actually the breadth.
Yeah. So looking more at like Q value and the breadth of the actual spores, not necessarily focusing on um, high and low length and width measurements.
Right.
Mm-hmm.
Yeah, and
That
also
makes
just the
sense.
average, because it's like you're going to have outliers, but there's generally an average that things fall within.
Mm-hmm.
Yeah, what do you do with Chylocystidia there?
What about them?
What do you do with them? I mean, I could I could tell you they look sort of like bowling pins, but they've got quite a range of shapes there and different sizes. And
Yeah, I
Yeah,
mean
what are
that's
those? Subcapitate and ligeniform?
um yeah, honestly, I have to look up the terminology every time I look into cystidia. That's something I can't retain. I mean, I would say they're pretty similar looking to cubensis. Based on spore size, it seems pretty similar in terms of the range of the size and the dimensions. Uh,
Yeah, but there doesn't seem to be any variation in the number of stegmata.
For now.
For now.
Because didn't they find some serolescens that have they're like bisporic? It was called Psilocybe bispora, I think.
Bisporous for
And they're
quite a
actually
while.
yeah.
Yeah, yeah. It
And
was
there's
like
like populations in South America, if I recall correctly.
the
It's like
the one we found in Mexico with Alan. We did that video. It's like a microscopy video, and
hmm
there's bisporic basidia in that one, and that was the La Montaña serolescens. So you probably have spores of that already.
hmm
so, what do you think about the stone-aped potential here? The Homo erectus following the proto-bison out of Africa as they pooped out their Psilocybe cubensis, most recent common ancestor with ochraceocentrata food. It's got potential, right?
Yeah, I mean, I personally was very entertained by his hypothesis. It seems like there's viable ideas that could hold water, though a lot of people, you know, poo-poo it, for
well,
lack of a better term.
maybe we just need to eat more Ocraceus and that's the secret recipe for like doubling our brains again.
That's the true thing, huh? Yeah.
Well, it would have been the most recent common ancestor, so maybe we can recreate that by hybridizing ochraceocentrata and cubensis.
yeah, that was what was interesting with the one phylogeny. Some of the polymorphic sequences seem to be behind the Ocraceocentrata and cubensis as far as like on the molecular clock analysis.
Hmm.
Oh, I missed that.
It's not on there. It's the some of the ones that I was doing with the cubensis Ocraceocentrata hybrids, sequencing a bunch of the F1s. Scott was actually just doing some validation of some of those sequences. We're going to put them on GenBank, so then people will be able to look at the polymorphic ones. But yeah, on the phylo tree, some of the polymorphic sequences almost end up looking like an ancestor rather than an offspring.
Hmm. I know the things that I've gotten that have been called Natalensis, but were supposed to be Ocraceocentrata. And if you sequenced them, you'd come up with Ocraceocentrata. But if you sequence the genome, then you see that there's cubensis in there too. You
hmm
got you got kind of
Yeah,
a
exactly.
distribution of both ITS types in there.
Plus polymorphic ones. So
Hmm.
yeah, the hybrid sequences that he's validating, I think a good bit of them were cubensis. Not too many of them were Ocraceocentrata, and then the other ones were polymorphic.
Yeah, I only got one Ocraceocentrata, like six
Seven
cubensis,
cubensis.
six or seven, and then five polymorphic, and it was all from the same culture, correct? Or
Well, it's
F1.
from F1 monokaryons from the same poultry, yeah.
Okay. Yeah. Um,
But that's also something interesting to look at with maybe nanopore from now on, so I don't have to isolate 20 monokaryons. I can just use a dikaryon and look at all the different consensus reads.
yeah. That way you can see the actual distribution of what's it called, OTUs within a single,
Hmm.
yeah, see if there's a dominant haplotype.
So I guess that's a project. We've already hybridized Ocraceocentrata and cubensis with different things, but maybe trying to find some sort of as close to um, like an origin cubensis as we possibly could. Would we have to like do some genomic testing on some different populations or whatever? But we could use that and hybridize it with Ocraceocentrata and try to find our ancestor through the polymorphic offspring from the F1.
I've been trying to get those two groups together, but it hasn't worked yet. Um, the ones at Maludi, Chuxiongensis, Pseudoacicorum natalensis, and Indica CF, which is probably Chuxiongensis, are in
Mhm.
with the cubensis ocraceocentrata. So I tried to do the bridge species thing, and that
Mm-hmm.
hasn't worked so far.
Mm.
Where I took the Pseudoacicorum natalensis hybrid F1s and then Ocraceocentrata cubensis hybrid F1s and tried to hybridize those together because the ITS difference I think is like 96.5%. And for the suboriginacean CF and the Griseo cerulean, their parents wouldn't cross and they're about 97%, but the Griseo cerulean and suboriginacean CF were able to cross. And I tested this with multiple different tetrads too. So it seems like the Griseo parents will not cross with the Griseo. I can't back cross it, and they won't cross with the sub CF. But I tried to use that logic of that like 97% difference in IT or 3% difference in ITS, 97% similarity to make that big of a jump. But it hasn't worked with those particular groups. But there's a lot of other species in that group that I'd like to play with. I don't even think really anybody has Maludi.
Have you
Some
thrown the F1s of that Pseudoasticorum natalensis at anything else, like the rest of the cubense section?
I tried it with cubensis. I tried it with Ocraceocentrata. It didn't work. I've done both for both. So I took Ocraceocentrata cubensis F1 monokaryons, and I tried to cross them with Pseudoasticorum by itself, and with natalensis by itself, and with Indica CF by itself. None of those worked. And then I did the reverse, where I took Pseudoasticorum, Indica CF, and natalensis, and tried to cross each of those, or tried to cross the natalensis Pseudoasticorum F1s with Ocraceocentrata by itself and cubensis by itself. And then I tried to cross their hybrids, and none of it's worked
Mm-hmm.
with multiple tetrads.
Hmm.
Well,
But they're
according
good
to
candidates for
this,
protoplast fusion.
Yeah. I was really hoping that bridge species concept was gonna work out. Ah, back to the drawing board.
I just don't think we see enough in that. Yes. What were you saying, Jason?
I was just saying, according to this figure in the paper, they're separated by 10 million years, plus or minus 5 million years, it looks like.
Yeah, those two groups. Yeah, that
Yeah,
was my, yeah. Between 5 and 20 million years was my guess, based off just kind of looking at it and extrapolating with that bar.
it's an awful long time.
Yeah.
I'm not sure that I necessarily agree with this, though. I mean, we've done other analyses that put this time frame about half of that.
Okay.
And there's some assumptions in their phylogenetic in their molecular clock that, you could impugn, like the Yule model and not having multiple outgroups kind of distributes all of the divergence evenly, but maybe it's not divergence between Agrossoby and Psilocybe, which ends up letting Psilocybe move pretty far back here. So I think that these are
Oh,
probably
to
all much younger.
what, more species to root the tree then?
Yeah, more species in the root. Because, yeah, you just end up making all of these branch lengths spread out evenly, more or less, towards the root.
Hmm.
But if
That
you
makes
were
sense.
if you had a
Maybe
lot of
different genera.
differences. Yeah, if you had more differences in agrosity, I think, then you could see how far the agrosity have diverged. Then it will, it will affect this root node here to some extent. I mean, it's also based on four genes, I believe, or three protein coding genes, three or four, and that might be biasing the data as well. I don't know. I just suspect that these are younger for various reasons.
Hmm.
Maybe like half this age for a lot of these nodes.
Yeah, they use TEF1, RPB1, and RPB2. If ITS was included, um, it would make sense that they might come out younger, right?
I think they used ITS in this as well, didn't they?
I think for
Uh,
this, they just pulled the proteins.
yeah.
There's
Oh, for
probably a
the
protein
clock.
model.
Yeah. Okay, I was gonna say in the paper they talk about the four, but yeah, maybe for this part, I'm
And
not
then
using
isn't
ITS.
isn't LSU also kind of a standard region to use for dating?
Yeah, but it wouldn't have fit into this analysis if this is a protein analysis.
Right.
Maybe you can combine them, but I think that they just didn't for one reason or another. Either it's not easy to combine two different data types, or the model would be too complex to include both data types. But
Hmm.
yeah, you can date with anything you want. The more data that you have, the better, but it's also harder to go through your calculations at some point. Like they'll do these analyses with like 1,000 genes. That way you have you have much more fine-grained information.
Yeah.
But I mean, it's also based on that one secondary calibration point there. And there's not really a fossil here that's dating it. It comes from a node in a previous work that had a fossil that was 100 million years old,
Hmm.
and one that was, like in the 80s of millions of years old, I think. So yeah, we don't really have anything to fix a date in this entire genus, and we probably never will. Takeaways?
Let's see. Yeah.
I
What
mean,
sort of takeaways do we have?
going back to what Julian said, it might have been before you like officially started, but people still calling it cubensis or wanting to call it cubensis. And for the most part, I'm going to assume these are cultivators who may or may not be, and this isn't like a diss, but taxonomically literate or, have their finger on the pulse. But, this is something to get used to. Like the sequencing of a type specimen can change, our understanding what a species is overnight. And, something we take for granted, like lion's mane, as far as I know, there's no type sequence of that. And if they do, if someone does that, I mean, the name for that might change. It might be, one of the other Hericium species. Um, and, there's plenty of cases with that, but most people typically don't bat an eye because it's a boring genus. Like, well, I think Satharella is cool, but a lot of people probably think Satharella is boring. They lack any like real charisma the way that Psilocybes and like Stropharia do. But once a species that someone actually cares about, um, the taxon name gets changed. It's like it ruffles their feathers.
Hmm.
Uh, and it's really nice to see that Bradshaw is cont continuing this type specimen work, and it'll be exciting when all these GenBank accession numbers go live and everyone can kind of dig into the data more. and use them in their own analyses.
Yeah, I agree with that. Someone recently asked me, I forget what they were asking about, and they were like, "Is it possible that could change?" And I was like, "Well, with taxonomy, it could change at any time for any reason, really. You know, species names and stuff like that."
Yeah.
Well, this work has legal and economic consequences too. Like, because Oregon says cubensis only, and so
Oh
all the
yeah, yeah,
gnats and
exactly.
hybrids gotta
Well,
gotta
I was
go.
thinking about that too. Hypothetically, if the cuben and they noted that they confirmed that cubensis is cubensis, but had cubensis come out differently, then there would be no available species for use unless some.
Hmm.
Does that make sense? Like,
Yes, that
Yeah,
would
if
be
yeah,
what we had
hilarious.
as cubensis wasn't cubensis, yeah.
yeah,
And then nothing's legal. They
Yeah,
already knew it was a ploy.
yeah.
yeah. The government was secretly like type sequencing stuff and just had it.
For
Uh,
anybody interested in the supplementary data from this paper, you can download it. There's a link. It's called data availability. It's under that bold heading, and there's a little link at the end of it, so you can look at all the microscopy, all the genome assemblies, the alignments for the trees, and everything. If you want to sift through all that data and see how the study was conducted, that's all available. Well, all right, guys. I want to thank everybody for spending the time with us tonight to talk about this. Scott, I think that point you made about the cultivation community, basically, I don't know what the right way to put it is, but just having to accept that this is how taxonomy works and this has changed. So we need to stop calling it Natalensis. We need to stop calling it cubensis. It is its own species. It's been
It's not AFF Nats either.
not AFF Nat anymore either. It's
Yeah.
yeah, it's got its own name now, so we need to start calling it that. As responsible amateur mycologists, it's the least we can do. So, once again, I want to thank my guests, Julian, Scott, Jason. Thank you so much for your time tonight, and hope to catch you guys in the next episode of Fungi Files.
cool.
Awesome.
Thank you.
It's been fun.
Yeah.
Thanks, man.